Okay, so I wanted to make a short video as possible over the activation of a naive B cell. So remember that when we're born, um, in the early parts of, like in utero and the early parts of our lives, our B cells will select which antibody they are going to produce, and we'll produce them. So you'll have a B cell with an antibody sticking off of it. Your body will double check all of those antibodies to make sure that they don't react against self, and if they do, they'll destroy them through a process called clonal selection. And then those antibodies, those B cells with their antibody, will be released into our blood, into our body, and will be there until they're needed. And those B cells that have never been used before are called naive, or sometimes you'll hear them called virgin B cells. Because they've never been used before, there's some extra steps that the body goes through to activate them before we start producing copies of those antibodies. We don't want to do all of that work and have it be done accidentally. So if we've never come into contact with that antigen before, then the B cell whose antibody matches that antigen will never have been used, and we'll have to do some extra steps of activation. So this video is about the activation of a naive B cell, one that has never been used before. Okay, so you come into contact with, some organism. So maybe put some bacteria that come in. You get a cut, you've got lots of bacteria that come in. Okay, so that bacteria then draws the attention of the immune system, and through the process of inflammation, all of the good immune system from our bloodstream is brought to the site. So we'll have phagocytes, we'll have B cells, we'll have all kinds of good helpful things, clotting factors. All of that will be brought to the site of the damage where these bacteria are. And so there's kind of two aspects that are going to go on. You're going to have your phagocytes working, and then you're going to have your B cells and your T cells that are in the blood that are going to come and see if their receptors match any of the antigens being produced or being seen in the infection. So let's first start with the phagocytes. So we have put most of our attention into the phagocytes, macrophage and dendritic cells. I'm just going to put macro and den. Okay, macrophage or dendritic cells both can do this role. These two phagocytes in particular are antigen-presenting cells, meaning they eat and then they take a piece of what they ate and they show it on the outside of them. So they have an MHC type 2 that will show a piece of the foreign thing that they ate. So let's show it presenting a little piece of our green bacteria. There we go. So, ta-da, that's what that macrophage just ate, that little green piece. Okay? Okay. Looking at what the phagocytes are presenting is a different group of cells that are drawn to the area through inflammation, and those are T cells. So in particular, looking at what the antigen-presenting phagocyte is presenting here on its MHC type 2, we're going to focus our efforts for the B cell activation in the type of T cell called a T helper cell. So T helper cells have a specific receptor on the outside of them called a T cell receptor, and that T cell receptor interacts with the particular antigen being presented on an MHC type 2. So not only does it have to be a particular antigen, but it's got to be an antigen being presented by a phagocyte in order to activate that T helper cell. So your phagocyte, your antigen-presenting phagocyte, is going to be showing a piece of what it ate, and the T helper cells will come along to see if they can, find the T helper cell that matches this new antigen that our body's never seen before. When that matching T helper cell has been found, then that T helper cell will get excited. It's never been used, and now it's used, and it'll start releasing a bunch of chemicals called cytokines or interleukins in particular. So we've got this T helper cell that has being used for the very first time. Its receptor matches an antigen we've never seen before, and it starts releasing chemicals and is very, very happy. Okay, so that's happening at the site of that damage or that infection. Also happening at the site of that damage or that infection, other cells that are drawn to the area are our B cells. So here we got our B cells. Okay, and remember our B cell produces an antibody that it, chooses. It's destined to produce this one antibody for its entire existence. And all of these B cells will come up and will check the antigens being presented at the site of the infection to see if any of them matches. When the one who matches the infection is found, the one whose antibody matches the antigen found on the outside of that organism, that will trigger this B cell to actually eat it, to actually engulf that organism and turn into a phagocyte itself, and present on its MHC type 2 a piece of the antigen that it just ate. So let's go back and put our antigen on our B cell. So here we've got a T helper cell that matches that green antigen. We've got a B cell whose antibody matches that green antigen. And because this is the first time that this B cell has ever been used before, it gets excited and it starts releasing chemicals just like the T helper cell did the first time it was used. So now you've got B cells releasing these chemical signals. You've got this T helper cell releasing these chemical signals, and that attracts them to each other. Okay, so the T helper cell is going to swing along and look at what the B cell is presenting on its antibody. And in theory, this T helper cell matching this green antigen should also be able to match onto the green antigen being presented by the B cell. And if that happens, then that tells the body that this green antigen is one that we've really never seen before. We have this T helper cell who saw it. We've got this B cell that saw it. It's really a real thing. It's really a real infection that's attacking us. So we need to respond. So if the T helper cell and the B cell match, then that tells the body that we need to set off a process called B cell expansion, B cell proliferation. And what's going to happen is that B cell is going to copy itself. And each one of those B cells will produce that same antibody that matches the antigen that just matched our infection. The difference with these new B cells that were produced is that they are not virgin B cells. They're not naive B cells anymore. They are B cells that have differentiated. They no longer require a T helper cell to activate them fully. So they're kind of pre-activated, skipping that T helper cell step. So part of these B cells will differentiate into a type of B cells called plasma B cells, or you'll hear them called just plasma cells. And these are the antibody producers. These guys will produce thousands of antibodies a second into our bloodstream. They are the little antibody factories. They produce antibodies and die. So they're usually short-lived, something like 2 or 3 weeks, basically the length of a typical infection, and then they die off. Okay, so they flood our bloodstream with antibodies and then they die. The other type of B cells that are copied, a much smaller portion, are called memory B cells. And these memory B cells live with us for a long period of time, decades, in fact. And the beauty of the memory B cell is that the next time that antigen is seen, so the next time that little bacteria pops it up in our body, what's going to happen is that these memory B cells can be directly activated just by bumping straight into that antigen. They no longer require the T helper. So when that memory B cell comes in contact with the matching antigen, it will set off expansion and it will make plasma cells and more memory B cells. Okay? And when that memory B cell comes into contact, it will also make more plasma cells and more memory B cells. So once it's a memory cell, it no longer requires the T helper. that makes it much, much faster. The T helper cell plus the activating of the naive or virgin B cell, trying to find the T helper, trying to find the one B cell whose antibody matches the antigen, that usually takes a week or so, somewhere between 5 and even 10 days sometimes to find the match. Once you're at that memory stage, you've got the memory B cells that don't require the helper anymore, plus you have more than one memory producing that same antibody. Now we're down to a couple of days to get the antibody production ramped up. So that's what vaccines do. Vaccines give exposure of the antigen so that we can produce memory to it, and we don't have this, week, 5 to 10 days of weakness and exposure, weakness of exposure where the organism is in our body, but our antibodies are not being produced at high levels yet, and we're completely dependent on our non-specific immune response at that point. Vaccine skips that and gives you exposure in a really controlled way so that when you actually come in contact with the nasty organism, you're at the memory stage and you only have a day or two before your antibodies come out at a very, very high force. And with each exposure, you make more memory, and so your speed with which you respond is faster, and the amount of antibodies you produce is more with each exposure up, to a point. But these memory cells last with us for decades. So you have, created this little army where every time you see it, your body responds bigger and faster. Hopefully that helps clarify the activation of the naive B cell.